Fondufe-Mittendorf, our alumni online, most importantly, our members of staff, academic staff, Professor Gumba, Dr. Ala, Dr. Okore, and Latuji and Ibrahim, and all the other lecturers here. The most important segment of this program are our students. Very lovely looking, amiable students for who we have organized this. This is the first in a series of lectures that is aimed at improving learning outcomes of students of zoology. There are a number of distinguished alumni of zoology outside of Nigeria and within Nigeria who have done exceptionally well with zoology as a course. The era of what course did you study is gone. What the society is looking for now is what you can offer. And believe me, zoology has the robust platform for you to spring from. It's a good place. Put yourself on. For you to spring from. So we are bringing practical examples of our graduates who have done well with zoology, so that this can encourage you and you can also link with them. It's also a good opportunity and a springboard for you to be able to advance your career in the different areas of zoology. As you know, there are 6 units in zoology: ecology and environmental biology, hydrobiology and fisheries, parasitology, entomology, cell biology and genetics, animal physiology, and all these units have an element that is related to them. And that element, that springboard, is molecular biology. Zoologists are breaking the glass ceiling so that we can open up your mind to the opportunities that are inherent in zoology, and know that zoology is a multidisciplinary and transdisciplinary course. It goes beyond the basics so that you would have better outcomes, and then we can all have a better template. So now we will invite Prof to give a lecture, but before then,
she has worked at leading institutions including the Modalities, Institute of Biophysical Chemistry, Northwestern University, and the University of Perturbation, where she advanced from assistant to professor. Her research investigates the impact of environmental factors on epigenetics, somatic biology, and disease, especially cancer. She has made key discoveries about how substances like arsenic alter chromatin structure and gene regulation. Her work has advanced understanding of histone variants, DNA methylation, and genome optimization, earning her numerous prestigious awards and grants, such as the NAIA Thomas Mackay Award, the University of Kentucky Research Professor Award, and the Charles Berkeley Research Excellence Award, as well as leadership of several major NIH and NSF projects. She is an active member of international professional groups, including the American Society for Biochemistry and Molecular Biology, the Biophysical Society, the Epigenetic Society, and the Society of Toxicology (SOT). She serves on the editorial board of boards of scientific journals such as NAR Cancer, NAR Medicine, Journal of Biological Chemistry (JBC), Oncology, and Scientific Reports, and is a reviewing editor of eLife. She also reviews grant applications for major funding agencies, including the NIH, NSF, and US Department of Defense, and advises the NIH for the nuclear program. She has given invited talks and keynotes at conferences worldwide, highlighting her recognition in molecular biology. She is dedicated to mentoring, having trained highlighting her recognition in molecular biology. She is dedicated to mentoring, having trained many undergraduates, graduate, and postdoctoral students who have gone on to successful careers. Professor Fondufe-Mittendorf has authored over 50 peer-reviewed articles in leading journals, significantly expanding knowledge in epigenetics, cancer biology, and environmental health, and continues to contribute globally through our research, teaching, and leadership. Ladies and gentlemen, with a warm round of applause, let's welcome Professor Yvonne.
Okay. So I'd like to thank all of you for coming in today, and I'm really humbled to actually be here and talk about my journey, scientific journey, as well as my career. And my hope is that I encourage a young girl sitting there, or a young boy sitting right there, and who's not understanding what to do next with their life in zoology. But to tell you that I'm indeed very grateful to the University of Ibadan, as well as to the Department of Zoology for giving. But before I go, I think this is something we always would say when we're UI breakers, UI, so we're still grateful for that term, or that slogan that we always use. Just being excited to be an alumni of the Department of Zoology. So today I'll tell you about some of my work that I've done, but before I go on, I'll strategize my talk in this way. I'll tell you who I am, I'll move to my career journey, my scientific journey, and I'll talk about one very exciting work that we have in the lab right now, and we're about to submit that for publication. So who I am, I was a young girl at 17 who moved from Cameroon, a neighboring country to yours, and moved to Nigeria, to Ibadan. Nigeria has actually given me a lot, the foundation it gave, because my dad at that time, there were no universities that catered for the English-speaking population. And since I couldn't speak French very well, my dad thought I should go to Nigeria. Unfortunately for me, my sisters were in Ibadan, and so that's where I came to Ibadan. And of course, it was the best university in Africa at that time. After finishing, I did my BSc and my MSc at the University of Ibadan. At that time, I actually thought I was going to do medicine. I was going to apply to UCH, but I didn't get admitted there. Maybe that was a good thing I didn't get admitted. I'm grateful for where I am right now, and I think I like the discovery rather than treating the patients. And like I said, Ibadan gave me that foundation that launched the next. Decided that, well, maybe that wasn't the place for me. I didn't want to work at that time. I thought I needed more to learn a little bit more, and rather than just start working. So I went to the University of Göttingen in Germany. I did a PhD there, and then I did a short postdoc at Max Planck Institute, which was just off the hill. I moved from there later on to Northwestern University in Chicago, where I became a research associate. It took me a while, to actually about 6 years to finish. But this phase actually was very important for me, and I'll explain why it was very important, because that launched my career, independent faculty career. I moved to the University of Kentucky, where I started there. I rose from the rank of assistant professor to do that within high-tier journals. I was able to get 3 NIH grants at a time. And just to bring your point here, in the US, when you're a faculty member and you're a research faculty member, you're supposed to actually, it's like a business. You're supposed to actually obtain grants so that you're able to hire people to work in your lab. So at every given time, I was very lucky and fortunate. And at every given time in a year, I was able to get about $1 million from the US, the postdocs, and high school students. So I trained graduate students, undergraduate students, and high school students. I'm a strong believer that if you give people the opportunity to learn, they will be able to take that and run with it. We need to be at the table and not sit at the side of the table. So I believe in us being active participants in research, and most of the time we're not given that opportunity. But I try as much as possible to bring undergraduate, high school students to work in my lab. That's with the Andel Institute. This is the 5th best institute in the US. So I'm indeed very grateful and humbled that I was able to have a lot of support and hopefully success. And that helped me, catapulted me to where I am today. Care of cell biology. And this role is important because it's given me a full round where I started off learning the ropes and eventually being in the administrative leadership. But I still hold a lab. So my scientific trajectory, I've always been excited to understand how your DNA contributes to who you are. And yes, we inherit that DNA from our parents. We're going to pass them up to our. So the 8 proteins here, 2 copies of histone H2A, And so you have 147 base pair of DNA wrapped around this, 147 base pair of DNA wrapped, a 15 base pair, 147 base pair of DNA wrapped. And it goes on and on and on. And that's your most basic packaging level. Eventually, that folds onto each other to form what is known as a 30 nanometer chromatin structure. It's further folded into what is known as loops. And if you look at your nucleus or your chromosome or your DNA during interphase, it looks like a mesh of ropes all tied into each other. And so this is what you see. But at metaphase, what you see is a chromosome. So that's your most condensed form. But if you're looking at your cell normally, you see meshes of ropes wrapped. So again, this has been quite a long time. And for those of you sitting there, for the students sitting there, you can also be able to be where I am now if given that opportunity. And I actually see that also I have a lot of professors that I said I had to be thankful for. Dr. Mosoro was my supervisor, Dr. Hassan, Dr. Mwahu, Dr. Odaigo, and so many others. And also Dr. Bakari, who was actually my peer that time. So I'm indeed grateful for that. And so that gave me the foundation to move forward. And for my PhD then I did try to understand how, when you have mutations on that DNA, how that could be repaired. So for instance, typically you would have in your DNA a cytosine opposite a guanine. But when there's a mutation, you have a thymine opposite a guanine, and that needs to be repaired by a protein. So I was able to understand how this protein, the TDG glycosylase, how that will be able to remove this thymine, insert a cytosine into it, such that during replication, you have the cytosine opposite a guanine that is making the new strand and not having a mutation, which could actually lead to diseases. Unfortunately for me, that paper was published in 2002, and that helped me now move to Max Planck Institute, where I did a short postdoc, but that was actually trying to move from there now to Northwestern University. So in Northwestern University, I go back to this slide, I've moved from the DNA, the double helix, where I'm looking at mutations. I'm trying to understand now how that DNA, being packaged into this as a nucleosome, how that can drive diseases. And the reason being that most diseases that people talk about, they look at the DNA and they say you have mutations. But how that DNA is packaged, if it's not packaged correctly, you could actually have regulatory factors that don't need to bind to that DNA. They can come in and access that DNA, and if it's not done properly, it can actually lead to diseases, and I'll show you here. So what I was doing for my postdoc was trying to understand this level. What histone PT and post-translational modifications that can actually help in opening up the chromatin or closing up the chromatin. I'll show you an example of the histone PTMs, H3K on histone 3, lysine 4, there's a trimethylation that is present there, and that helps in opening up that nucleosome. On this other methylation, which helps in closing up that nucleosome and your chromatin, repressing gene expression. So all of this that I've told you, they constitute what is known as the epigenome or epigenetic regulation. And so the question is that why do we care about epigenetics? I actually like this Time magazine article which says why your DNA is not your destiny. Yes, you're going to inherit that DNA from your parents, you're going to pass that on to your offspring. But they go back and answer, it is this epigenetic marks that tell your genes to switch on, to switch off, to speak loudly or to whisper. And so the question typically when I teach my students in class, I ask them, if you have the same DNA and your DNA makes who you are, which is true, but yet you have your eyes, your ears, you have your liver, you have all these other organs in your body. If I commit a crime and you take my blood or my saliva, that DNA is unique to me. Yet I have all these other organs in my body. How is that possible? Again, it's the epigenetic marks that tell which genes to switch on or off, which genes to speak loudly or to whisper. So for instance, in your eye, you have the same genes in your eye that you have in your liver, yet your liver is not found in your eye. So what is happening is that those genes that are supposed to make your liver cells or are important for your liver cells in your eye cells, they are turned off. and only your eye, the genes that are important for your eye, are turned on. And so those parts are open for those regulatory factors to come in and read that information. And so why is it important? During development, you're constantly being exposed to cues, what your mother is drinking, what environment you are in. That would induce epigenetic effects, that would induce a phenotype. Indeed, so many phenotypes, changes in growth, reproduction, cardiovascular function, body composition, metabolism, appetite, food preference, behavior, neural function, stress responses, longevity, all of those, epigenetics shows to play a role in that. That could lead to a healthy individual or a diseased individual later on in life. Later on in life, what we get exposed to, what we eat, what we drink, how stressed we are, that also would send out cues. We could induce epigenetic effects, we could induce a phenotype, and it could lead to a healthy individual or a diseased individual later on in life. So my lab seeks to understand how, when we're exposed to environmental toxicants, how that could lead to diseases. And the one I work on is arsenic. Indeed, I'm a basic chromatin biologist, and when I started my lab, and so this is one of those funny things, when I started my lab, I wrote a grant to the National Institute of Health. When I sent it in, that grant was not, they didn't even look at it, and I felt so bad, I felt I was so stupid, I don't understand what I'm doing. But the program officer was very nice, and he said, well, Yvonne, you're actually playing with the big guys, and it will be interesting for you to come in and sit at study sections so you can also review grants and understand how grants are written. Which again, throughout my career, I've had people who've helped me at every stage in my career, and for all of those people, I'm indeed very grateful. And so NIH had a call for chromatin biologists to do toxicology, or toxicologists to do chromatin biology. I'd never done toxicology, and I kind of hated toxicology because my idea for toxicology was that people would just go into the environment and measure the levels of the toxicant that are there. But I've always been interested in knowing the how and the why. And so I had a colleague, the program officer said, you're actually what we're looking for, but you don't have a toxicology background. So I sought for a toxicology collaborator, and fortunately for me, she was working with arsenic. Never heard of arsenic before, or I'd heard of it in movies where a spouse is trying to kill the other spouse by lacing their drinks with arsenic. That's why I heard of arsenic. But now I understand that arsenic is ubiquitously found in the Earth's crust. So you and I are constantly being exposed to arsenic at low levels. And it's been shown that even chronic levels of arsenic, low levels of arsenic, cause cardiovascular disease, diabetes, cancers, and different types of diseases. And you're probably wondering, where do we get that from? And like I said, it's ubiquitously found in the Earth's crust. It's found in water. It's found in the food sources we eat. You and I, if we dig the soil, because it's found in the soil, we're exposed to that. But we're exposed to low levels. Also, those who work in coal mining are exposed to that. Indeed, those who work in, like for instance, those who go to war and they bomb the ground, that soil, that dust that comes out, we're exposed to that. It's actually found in fossil fuels. So driving the car in cigarette, we're constantly being exposed to that. So the question is that we cannot actually eliminate that. How can we find something that can actually protect us from this arsenic contamination, or what we have right now? You might say that doesn't bother me, but this is what the New York Times Magazine said in 2022. Because of changes in climate, goes to miles and miles away. And they actually said we're potentially having an environmental nuclear bomb. So this is happening in so many places. So you and I are constantly being exposed to that. Indeed, when I was looking at, because I wanted to bring this home, I looked at a publication from a professor in Ibadan who published this paper in 2014. And he showed that the well waters in Ibadan actually have very, very high levels of arsenic. Indeed, during the rainy season, because they're going to have micromolar of arsenic, that's indeed very, very high. And I'll explain why I say so. We have it everywhere. So this is in the US, where the data is readily available. They show that that high level of arsenic actually correlates with high incidence of cancer. Is shown here. The well water has about 50 microgram per liter. That's a lot lower than what we have in Ibadan, the well waters in Ibadan. And that's about 0.5 micromolar. In other places, we have about 220 microgram. Indeed, that's also high. But what we know is that arsenic does not directly mutate DNA. So you will not go in and see a strand break, or you're not seeing those DNA mutations in your DNA. So how is it driving carcinogenesis? So my lab thinks that what it does is that, so what we did then was we took cells, these to be epithelial lung cells, and we exposed them to arsenic, and we collected, as we're going through, we collected the cells. Because what I like is I just don't want to look at all or nothing, so no cancer versus all cancer. I want to look at the process. I want to understand how the cancer process is taking place and how epigenetics can be driving that process. So what I did was we took cells, we treated them with 0.5 micromolar of arsenic. Remember, this is the level that is found in the well water in Kent County where I live. And so you're moving from non-exposed cells right here, and you're moving through to exposed cells and then tumorigenesis. So what we did initially were, after exposing the cells to arsenic, we would measure migration. And what we'll do is that we'll put them in this little well right here. We'll put a chemoattractant at the bottom. And if the cells are migratory, which are actually a hallmark of cancer, they'll move from this level up here and move to the bottom. And we can actually measure how migratory they are. What we saw as we go through the weekly exposure with arsenic, we see that as we're going on, the cells become very migratory. You see from non-treated, not many cells migrated to the bottom. But as you go through the weekly exposure, we see high migration potential of those cells. And what was interesting for us at the time, we thought we made a mistake, but we've done it over and over. So after week 17, we moved to week 28. We actually see that the migration potential reduces. Indeed, this is the hallmark of cancer, where once you have this propensity to drive cancer, these cells migrate to other places. You would hear somebody say, I have breast cancer, now it's somewhere in the leg or it's somewhere in the bone. It's migrated to that region. So what we're seeing is that the cells become more migratory, and as they find an environment that is really good for them, that migration potential reduces, and they're able to invade that place and actually build up a tumor. And indeed, we're actually able to see that at week 28. We see that there are a lot of colonies that are formed, and they're more invasive. They don't migrate a lot, they're more invasive. And if we inject them into nude mice, so the way you can test how a cell is tumorigenic is that you can actually take those cells, put them, inject them into a mouse, and if they grow tumors, that means those cells become tumorigenic. And indeed, we're able to see that at the late stages of arsenic exposure in the cells, those cells become tumorigenic. And what we're seeing is epithelial to mesenchymal and back to epithelial. The epithelial state here is very different from the epithelial state here, because here, when you're just starting, the cells have no carcinogenic phenotype. However, at this other epithelial stage at the end, they now are invading and they're able to grow tumors. So there's a difference in the type of epithelial that you're talking about. But the process in cancer is always epithelial through mesenchymal and back to epithelial. So I'll talk to you how we try to understand what are the genes that are actually driving this process. So to answer that, we do what is known as RNA-seq. So you take your cells, you take out your RNA, you digest the RNA into small pieces. You actually would put adapters to that RNA. You could actually reverse transcribe them into cDNA, complementary DNA, and you can actually sequence. So when you're sequencing all of the RNAs in your genome. And so if you take that and you map them back to your genome to find out where these RNAs are coming from, you could actually found, this read will tell you, if you have many reads here, it will tell you that RNA or the genes were highly expressed. If you have very low reads coming up, you would say that gene was lowly expressed. And so you can compare that from arsenic exposed cells to cells that are not exposed to see what are the genes that are turned on or off and what are the genes that are helping to drive this process. So again, just to show you what we did, so this is all the RNAs that are found in your body, and we look at those genes that are differentially expressed. So in red would be the genes that are highly upregulated, and in blue would be the genes that are downregulated. We were able to see about 3 clusters where they have slightly different patterns, but all the upregulated are oncogenes, those are cancer-driving genes that are upregulated, and the genes that were downregulated are tumor suppressors. So those are the genes that we need in our body to actually protect us from tumors. And we see that those genes are downregulated, suggesting that when the tumor suppressor genes are downregulated, it means that the oncogenes, nothing is there to actually protect us, the oncogenes just go up. And so this is with arsenic-mediated transformation. So today I'll tell you about one example in the lab that we were very excited about, and that's to understand how arsenic could directly change the chromatin structure and how that could change gene expression. And I'll talk about a story which we had published before, but we've now worked and understood now the mechanism by which arsenic could be doing that. And I'll talk to you about a 3D genome rewiring and how arsenic is actually changing in how you package that DNA to actually drive cancer. So I've shown you this in a different form, where I say you have your double helix, you have your beads on a string form of chromatin, you have this folded again into loops. And again, I'm just showing you more details of this packaging of the DNA. You have the loops formed, but you could have several of these loops formed into, folded into what is seen a subtle change in the 3D genome. We could also look at compartment B versus compartment B. We start with a strength of about 2.4 and we move to about 3.8. If we look at the topology, from encroaching a repressed topological associated domain. And so what we're trying to see, do we change those topological associated domain? And so what we're measuring is whether that topological associated domain A can actually be reverted to topological associated domain B. So if you look at this and you're moving from non-treated samples, we see green to this region right here. Whereas we move from non-treated to week 3, to week 8, to week 17, we start seeing, and week 28, we start seeing a lot of these red regions, suggesting now that we are flipping an active topological associated domain to a repressed topological associated domain. We now looked at loops, because these are the different hierarchical folding of the genome. We looked at loops, so enhancer to promoter. Indeed, we're able to show that there are changes in the loop strength. So this is an example of a cluster 1, where we see we move from a low loop strength to a high, to a high, to a low, and to a low. If we take where that loop strength is found in the genome, in non-treated cells, and we're moving towards the cells where we have full transformation, and in blue we're losing CTCF. That correlates very nicely with open chromatin structure. So what I'm saying here, we do ATAC-seq, where you can actually measure how open that chromatin is. We see in red, those are open chromatin regions. We start with non-exposed cells and we move towards the more carcinogenic state. And we see that again, the pattern looks very similar to CTCF, where we are losing the open chromatin region. We are also losing active histone marks. Like I told you, the histones get modified depending on whether those histones, you have active marks or repressed marks. Those histone modifications can actually close up the DNA or open up the DNA. And indeed, we're seeing that right here, where we see also, because we're losing CTCF, we're also losing that active histone mark. And also, we're losing gene expression in red to blue. We're losing gene expression during that stage. But what was exciting to us is that we're actually gaining a repressed mark And this repress mark means that the genome is actually being closed up. And this was exciting because, in fact, we now think we've answered the question in the field, which would actually change what people have been thinking, which is that the classical model is that CTCF binds, you have a topological associated domain A, which is active, and CTCF actually acts as an insulation, blocking this topological associated domain from a repressed topological associated domain. And so in the field, people have tried, if you lose CTCF, the idea is that you would lose this insulation such that you can have merging of this topological associated domain A moving into topological associated domain B. But what we've seen is that in our case, arsenic is actually preventing CTCF from binding to the weak CTCF binding site and driving carcinogenesis. And how it's doing that is that it's a localized region where it's actually in local chromatin 3D structure. It's actually removed the insulation that would actually have occurred, and it's actually allowed a protein known as PRC2 to actually write in this H3K27 trimethylation, which is a repressed mark, and you're having repression that way. And you can imagine that if, again, this is a tumor repressor, just by doing this, you've actually reduced the ability of that cell to counteract tumor progression in the cell. So the question for us is, I know most people will ask that question, how does arsenic do that? So this is a structure of CTCF. CTCF has 11 zinc fingers. And so zinc finger proteins, they have this zinc finger that helps them hold onto the DNA and read the information that is on the DNA. The CTCF has 11 zinc fingers. 10 of them are C2H22. So 2 cystidines and 2 histidines holding, coordinating that zinc ion. There's only one C2H1, so 3 cystidines and 1 histidine coordinating the zinc ion. Previous studies had shown that those proteins that have C3H1, arsenic can come in, displace that zinc, and when it displaces the zinc, it inserts itself into it, and that protein is no longer able to bind. So we think this is how arsenic is doing that to CTCF. Indeed, with CTCF having these 11 zinc fingers, not all of them engage DNA at the same time. Zinc finger 1 to 3 would help scan the DNA. When it recognizes that there's a binding site for CTCF zinc finger 4 to 10 would go in and hold onto that DNA. And zinc finger 9 to 11 would kind of bind to the upstream motif and stabilize CTCF binding. And so what we're saying is that if this is the 11th zinc finger and you have a weak CTCF binding site, CTCF doesn't like to bind because that's not its core sequence. However, it needs to be stabilized by a zinc finger 9 to zinc finger 11. If, for instance, CTCF is now disrupting that zinc finger 11, it means that that stability of CTCF binding to its site is no longer present. Indeed, we're able to do mass spectrometry. Again, this is the 11th zinc finger of CTCF. If we use that protein, dialyzed it in a buffer that has arsenic, what we saw with wild type is that CTCF is able to, arsenic is able to go into that region and actually displace the zinc from that finger, because we see more arsenic to zinc compared to when you don't have arsenic. On the other hand, we then decided if it's true that it's this 11th zinc finger that is targeted by arsenic, we mutated that 11th zinc finger to C2H2, which would typically not be a target of arsenic. And indeed, we're very excited with that because we did see that arsenic is no longer able to displace zinc, and so we have a reduction of arsenic in that zinc finger. What we did then is, how important is this zinc finger? How important is arsenic? How important is CTCF in driving oncogenesis in the cells? What we did then, I'm showing you here the gene expression patterns when you have your cell types and you have arsenic in it and you have wild type CTCF. We see that it's actually driving a lot of KRAS signatures which are all oncogenic signatures and help to drive cancer. When we did RNA-seq also with this mutant cell line now that has CTCF, that has C2H2 instead of C3H1, which should actually inhibit CTCF binding, which should actually inhibit arsenic binding. What we saw was this. I hope you can actually see what I'm saying. If you compare the first line right here and the line right here, you're actually having a dampening of the effect of the gene expression of the oncogenes. So what we're seeing is that in the mutant cell line, you treat them with arsenic, they're not actually turning on or off these genes that are actually driving cancer. We're actually seeing a dampening of the effect. I show you an example of one of those KRAS pathways where you see in the wild type, you see those genes are very highly turned on, or those genes that are highly turned off. All of those are oncogenes depending on the gene expression pattern. If we look at these genes that are now differentially expressed in the mutant cell lines, they are not significant, which means that they're not differentially expressed. So we're very excited to see that CTCF, that zinc finger, seems to be playing a role in how arsenic is driving carcinogenesis. Again, if we look at how the actually cells migrate, in wild type, what I've shown you at week 17, where there's the highest migratory potential, we see that this wound here is healing, it's closing up. And then at week 28, which I've talked to you, that's the most invasive state, we're seeing that that wound is actually open up. That's in wild type cells. In the mutant cell with the CTCF, we see that we didn't see any changes in the migration potential. We also did not see changes in the colonies that are formed. They were not able to form colonies, which means that they're not invasive. And if we injected them into mice, they were not able to form tumors, whereas the wild type in red was able to form tumors. So this suggests to us that that 11th zinc finger of CTCF is actually very important in driving arsenic-mediated carcinogenesis. Well, is this all about CTCF? The question you would ask is that, well, there are other proteins that have zinc fingers that are also of C3H1. But what is interesting is that there are about 50 to 60 proteins that have C3H1. Again, the African child, and again, I realize that a lot of people in my lab are not Africans, but we know about the visa issues. It's not easy, but that's not an excuse. How would you be able to link up with me? I could actually send my email address, or you can actually find that, and we could talk about that. The difference here when students are coming to work in a lab is that typically you don't come in directly to a lab. You come in through a program. So the grad school would actually look at the student's application, and they'll also interview the students. And then there's a selection process. So if you're selected, then you come in, and the first year, you actually are just doing classes, so you don't go to a lab. And your second year, you go in for rotation. No, sorry, your first year, towards the end of your first year, you do rotation. You rotate in several labs. And then you can choose the lab where you want to go to. But typically, there's no way you can come directly into my lab, so I don't have that control of you coming directly to my lab. You must come through that program. If you're admitted and you like my lab, then you can come to my lab.
Thank you very much. Please, any other questions?
I don't have this background of which you are talking about. That actually is association. Graduated in this university in New York, in 1988. But I saw this on the social media and I said, I want to be there.
Sorry, you're breaking off.
I'm attracted seriously by the topic of this lecture.
Okay.
And despite the fact that I don't have this background, I have been able to follow you through and through. Let me tell you what I have gained. I have gained that zinc, basically, I have gained that zinc supplements reduces the propensity to have cancer. And this cancer will be triggered by arsenic I put it as injection because I don't know what to do. And I also think that the earlier you start using zinc supplements, the better. It will retard the growth of cancer in the body, or it might actually stop it entirely. Now, my worry is, if we have this ingenious way of addressing this big problem that we have in, let me say, Nigeria and Africa, how do we get these things across to the laymen? Because I know that it's only the rich people that have been treated for cancer in this country. Very few cancer centers, and they are quite costly. I have lost 2 people to cancer, and they are quite costly. They are very draining. So how do we get it readily available and cheap to majority of the people? Laboratory exercise a different thing. Practical usage in day-to-day life is what I'm asking. How do we get that done? Thank you.
Thank you for the question. So I think I try to push yet that if we can actually test that in people, I might push for that. We haven't tested that in people, we've just tested them in lab animals. And so I want to caution for you not to jump into that. But what I'm saying is that we all take zinc supplements one way or the other. It's not going to be like I'm pushing you to take another type of medication. I'm just asking you to take the zinc. And even if you don't know that this is going to help you cure cancer, why not do it? We eat a lot of food sources that have zinc, and so give it a try. I have not tested that in human beings yet, and so I'll caution for you to tell people that some professor told you to take zinc. But we take zinc supplements all the time. So for me, I look at it like it's nothing different from what you do. And so if that's going to help you with some heavy metals, actually chelate some of those heavy metals in your cells, that's fantastic to do. And how do you caution the layman? Well, maybe zinc supplements in Africa might be expensive, but we have food sources that are rich in zinc, and I think people take that. So my suggestion there would be, take the food sources that are rich in zinc.
Thank you, ma'am. Okay. There are huge class of proteins that use zinc to stabilize their stock, and that is very important, which is very important in gene expression. So I'm asking, is there a way, I don't want to call it a structure, I want to call it like a reduction in the time the lab work. Is there a way you can reduce the huge amount of work done to get the outcome by using computational tools like statistical methods to get the outcomes? Just like the precursor to really validating these things in the lab.
And one of the epidemiologists actually said, oh, you know, there are actually a lot of people who are zinc deficient, and wouldn't it be great to actually find out whether these people have more cancer? And while they were going back and forth and someone was arguing, someone actually said a lot of those people live in arsenic, high arsenic areas. In fact, in Bangladesh, where arsenic is really high, in the soil where they do rice farming, a lot of people are zinc deficient, and they've seen a lot of skin cancer there. But there, I actually also think we have to also caution that, because their levels of arsenic is really, really high, and even those who are not zinc deficient are also having skin cancer. So I think your question would be, how do I validate it in humans? I would have to find out, because again, when I say arsenic exposure in in the soil, it's actually a combination of arsenic and other heavy metals. So while I'm looking at only this, there's cadmium, there's chromium, in other places there's lead. And so with this one, it gives me that drive to look for, is this the same thing happening with cadmium and chromium? And kind of combination of all of this drive the same. Yes, it's a lot of work, but I just don't like observations. I like to find the mechanism by which it's doing that, because by me knowing that zinc plays a crucial role, it helps to actually validate those if I'm seeing more with zinc deficient people. I hope that answered your question.
All right, thank you very much, everyone. With a round of applause, let's welcome back our head of department, Professor. And I hope you have learned one thing or the other from this interaction. When we were planning this less than 2 weeks ago, we decided that we were going to engage on our platforms. I posted it on my Facebook page. We posted on LinkedIn, and the link and the venue and all. I am very happy to have you, sir, attend, because that's what we mean by town and town interaction. And I believe that this is the first of many series. The next series we're having in May will be a physical day. Our speaker will be physically here. He's a zoology graduate who is now a very big man in Total Energies from zoology. And the good thing with this is that it not only opens your mind to the opportunities that are out there, it also gives us leverage at the department to look at our students and see how we can place them. Collaboration with those that are out there that can assist. I want to appeal to you, especially our undergraduate students. Last year, I did interact with you and I talked about certification courses, which was posted on your page. For you to get international collaboration, there are some things that need to be on your CV. One of them is certification courses that you have done. It shows that you are increasing your knowledge base, and that makes you more competitive. Some of those courses are for 6 weeks, some are for 8 weeks, and they are free. Please go back to those links, try and do those certification courses. It not only makes you a better zoologist, it also opens up a lot of opportunities, because when you go and do online certification courses in the University of Kentucky, for example, and you do well, by the time you are connecting with a professor who is part of the courseware, he will remember you that you are in his course cohort, and that can open up a lot of opportunities for you. We want our zoology graduates to raise their head up high. So just like what we did for the plastic pollution initiative in UI, we have over 130 students here. How many of you have LinkedIn page? Raise up your hand, please. I want you to try and take some pictures from this event. Write your thoughts and post it on LinkedIn. Hashtag zoology. We have Department of Zoology on LinkedIn. Hashtag UI. Hashtag Department of Zoology. Write your thoughts. On Facebook, do the same. You know what we are doing? We are promoting the discipline of zoology. So people don't think that zoology is just about those who work in the garden or those who teach. And then you can get a lot of people interested. When you go online and you say, what's the prospect of zoology in Nigeria? It's zero. The people that can build it are here. You and I need to build zoology into an enviable discipline that a lot of people will find very interesting. So just like we did for the plastic pollution, please, students, go on your LinkedIn page, go on Facebook, go on Twitter, write about this program, tag us in zoology. We'll also open a Twitter account. That way we can make the presence of Ibadan zoology as a premier and preeminent department of zoology in Nigeria and in West Africa. So thank you for coming. And then let's thank the National Association of Zoology Students for the cooperation. The postgraduate students of the department, you are appreciated for being here, and I hope you all enjoyed it. Yes, ma'am. Sir, do you want to say something to us? Just for 2 minutes before we leave. Because
to the Department of Zoology at the University of Ibadan. Incidentally, I noticed that this is the first Distinguished Alumni Lecture. We are all blessed to be here. I hope you know that so much has been passed across within these 2 hours that we have sat there. My prayers is that we will pick little bits of things from what has occurred here, not only from the lecture, but from the interaction with interface and so on, and the soft cells that the head of the department has passed across to us, and we will be able to make a better person of ourselves. We will be able to make a better representative of the zoological of the department. And we will also be able to present ourselves as worthy ambassadors of the University of Ibadan, wherever we find ourselves. Ma'am, once again, I want to thank you. I actually saw this on the WhatsApp page of the Senior Staff Club.
yes, I posted it here.
Yes. And I said to myself, if there is a place I have to be at 2 o'clock today, this will start. So, Prof, I thank you so much for the opportunity to gather me here. And Prof, I thank you so much for establishing this. And I'm very glad that you are saying that this is going to be the first of the series.
Hopefully, maybe next year I could do that. And actually meet some of you in person. But also, I'll tell you, the students sitting there, that as the others have been given here. And for me also, I think being able to sit here, so many people walked with me, and they helped me get to where I am. So reaching out to people online and, just asking what they're doing, whether they can be helpful, whether you can even do an internship, what is available. There's so many things that are available. I'll give an example. I used to sit in the admissions committee in my institute, and I would see a lot of Ghanaians applying, and I'm wondering, I know last year they had one Nigerian from Ibadan who did not make it, but I'm asking, what's wrong that we all cannot apply? You don't know if you have the strength. So please reach out. There's so many schools that are available in the US that are looking for people. And if one person comes out, I've done my job, right? So please reach out if you really need help from me. I could read some of the letters if you want to. I'm just hoping that more of us sit at the table to make those decisions. Thank you. Thank you so much for your attention.
We hope that this will be the beginning of collaborations. We're talking about a lot of things. A couple of years ago, we missed out on a batch of equipment that she was giving out because she was moving from one lab to another. And she had the opportunity to give out a lot of high-tech equipment that we could have gotten here. But it's never too late. We'll keep interacting with you. And she has also made some donation that will soon come to the department. So can we give her another round of applause? What Professor Vaughn has shown you today is the possibilities that are inherent in hard work. And I hope that this will be the beginning of rethinking zoology for you. Prof, we thank you very much.
Month of April. Thank you very much, ma'am, for the idea, for the support, for the guidance, for the refreshment. All what you do get this afternoon was provided by her. Thank you very much. So, on behalf of advisors, Professor Show, we want to say a very big thank you, sir. Professor Awokode, we want to say a very big thank you, ma'am. So, our lecturers, Dr. Ala, Dr. Anane, Dr. Topo, Professor Alake, Dr. Latunji, Dr. Ibrahim, Professor Oruka, Dr. Oli, Dr. Taiwo, Dr. Koka, we want to say big thank you, thank you very much. To all our postgraduate students, PhD students, master students, we say a very big thank you for honoring our call. And also to my fellow NASA, thank you very much. The national anthem, please.
Ibadan.